It is February 7th, 2026. I want to start by asking you to just mentally rewind the clock a little bit, about a decade. Go back to, say, 2015, maybe 2016. Do you remember the absolute hysteria when CRISPR first really broke into the mainstream consciousness?
Oh, it was unavoidable. I mean, it was everywhere. It was on the cover of Time, The Economist, Wired. It wasn't just a science story, you know?
No, not at all.
It was a full-on cultural moment. We were talking about rewriting the code of life.
Yes.
And the narrative was that we were like moments away from curing every single genetic disease and, of course, the inevitable designer babies.
The designer babies, of course.
Yeah.
And then we waited. For a long time, like if you're a layperson like me, it felt like the technology was just stuck. It was perpetually in that promising potential phase.
Right. It's always 5 years away.
Always 5 years away. But going through this massive stack of research you sent over for this week, and I'm talking specifically about the clinical readouts from late 2024 and all through 2025, it really feels like the dam finally broke while we weren't looking.
It did. It absolutely did. But I think the nuance that most people are missing, and this is what we really need to dig into today, is that the technology that is actually curing people today, right now, here in early 2026, it isn't really the same CRISPR we were all talking about back in 2015.
That was my single biggest takeaway from the reading. We're not really using the molecular scissors anymore.
Yeah.
The sources are calling it CRISPR 2.0 and even 3.0. We've gone from scissors to, what's the new analogy? Pencils.
Pencils and word processors. That's the analogy that holds up the best, technically speaking.
Okay.
We've moved from what was essentially blunt force trauma, breaking the DNA and just hoping for the best, to this elegant, precise chemical rewriting.
And the reason we're doing this deep dive today is that the clinical data backing this all up is no longer theoretical.
Exactly. We have real human beings walking around right now with chemically altered livers, with new immune systems, who have been cured by tools that frankly didn't even exist when that first wave of CRISPR hype happened.
So our mission today is to unpack these two major leaps forward. We're talking base editing and prime editing.
Okay.
We need to understand the mechanics without needing a PhD, hopefully, and then really look at the actual clinical trials. But before we get to all the shiny new toys, I think we have to talk about why the old CRISPR, the classic Cas9, the one that won the Nobel Prize, why it wasn't the magic bullet we all thought it was.
It's not that it wasn't a magic bullet. It's just, it was a very messy bullet.
Explain that, because the headline was always precision gene editing. That word precision was everywhere.
And it was precise in finding the location. That's the key distinction. That's the CRISPR part of the name, the guide RNA.
Okay.
Cas9 is absolutely amazing at using a guide RNA to find a specific address in your genome.
Yeah.
I mean, it can scan billions of letters of DNA and find the exact 20-letter sequence you want it to find. Incredible.
But what happens next is the problem.
But once it gets there, its only move is to cut. It makes a double-strand break. It snaps the DNA ladder completely in half.
And I'm going to assume the cell doesn't particularly like having its hard drive snapped in half.
It hates it.
Yeah.
It absolutely hates it. It triggers a cellular panic response. You have to understand, from the cell's perspective, a double-strand break is a 5-alarm fire. It's a signal that it is dying.
So it scrambles the emergency services.
It does. And the primary way a cell fixes that kind of break is through a pathway called non-homologous end joining, or NHEJ for short.
Which, according to my notes here, is stochastic, which is a very fancy scientist word for random.
It's completely random, right? Think of it as an emergency weld job. The cell finds the two loose ends of the DNA and just jams them back together as fast as it can to keep the chromosome from literally falling apart.
And I imagine some pieces get lost in the process.
Oh, yeah. In doing that, it almost always deletes a few letters, or sometimes it accidentally adds a few random ones. It creates a scar in the genome. They're called indels insertions or deletions.
The analogy that made it finally click for me was the encyclopedia concept. If I want to remove a sentence from an encyclopedia, well, tearing the page out works. The sentence is gone.
Exactly. That's what CRISPR 1.0 was great at, knocking things out. If you want to break a gene that's causing a problem, like, say, a broke that shouldn't be there, scissors are perfectly fine for that.
But what if you don't want to delete a whole sentence? What if you just want to change one word, change the word bat to cat?
Tearing out the page doesn't help you there, not at all.
No.
And in fact, we've learned over the last decade that it's actually worse than just being unhelpful. Making these double-strand breaks is inherently dangerous for the cell. It can cause what are called chromosomal translocations.
And that's where, what, the top half of chromosome 4 gets accidentally stuck to the bottom half of chromosome 12 or something?
Essentially, yes. If you make cuts in two different places at the same time, the cell's messy repair system can get confused about which end goes where. And that is a hallmark of cancer. Plus, cutting the DNA triggers the p53 response. You might have heard of p53. It's called the guardian of the genome.
Right.
If p53 sees too much DNA damage, its job is to tell the cell to commit suicide, to undergo apoptosis. So standard CRISPR is literally toxic to some of the very cells we want to fix, like precious hematopoietic stem cells.
So to summarize let's call it the Stone Age of 2015. Great at finding the spot, but the actual editing process, the repair, was messy, it was risky, and it was really only good for breaking things.
Correct. Right. Which brings us finally to the first big evolution, the molecular pencil.
Base editing.
Base editing.
This came out of David Liu's lab at the Broad Institute, and the fundamental shift here, the whole philosophy, seems to be the decision to just stop cutting the DNA entirely.
That was the entire design philosophy. Let's not break it. They took the Cas9 protein, and they broke the scissors. They deliberately mutated the protein to turn it into something called a nickase, or NCas9.
Nickase.
So instead of snapping the ladder across both rails, it just it nicks one side, just one string.
So no panic response.
No panic response from the cell. No p53 trigger. The fundamental structure of the DNA remains intact.
Okay, but if you aren't cutting, how are you editing? If I can't cut the bad piece out, how do I change it?
Chemistry. This is the absolute brilliance of it. They fused a second enzyme to this hobbled Cas9, an enzyme called a deaminase.
Deaminase. Okay, let's unpack that word.
A deaminase is an enzyme that performs a direct chemical reaction on the DNA bases themselves. It literally rearranges the atoms on a specific letter to turn it into a different letter.
Like using a little dab of whiteout and just writing a new letter over it.
Chemically speaking, that's a perfect analogy. It's a chemical conversion.
So it can turn a C into a T.
Yes, that's what a cytosine base editor or a CBE does. It chemically modifies cytosine C into uracil, which the cell's machinery reads as a thymine T.
And there's another one.
There's an adenine base editor, an ABE, that does the same thing but for A's. It converts adenine A into a molecule called inosine, which the cell reads as guanine G.
Okay, but wait. Let me try and recall my high school biology. DNA is double-stranded.
Right.
If I change a C to a T on the top strand, don't I now have a mismatch with the G that's on the bottom strand? Because C pairs with G, but T pairs with A. So now I have a T paired with a G. That's a mismatch, right?
You do. You absolutely do. And that is where the nick comes in. This is the really clever trick.
Ah.
The Cas9 nickase nicks the unedited strand, the one with the original G on it. The cell's repair machinery sees the nick, it sees the mismatch, and it assumes the nick strand is the damaged one because it's, broken.
So it fixes the wrong strand.
It repairs the bottom strand to match your new edit on the top strand. It erases the G and puts in an A to correctly pair with your new T.
It tricks the cell's own systems into saving the edit. That is incredibly clever.
It co-opts the cell's own mismatch repair machinery. And the beauty of it is, again, no double-strand breaks, no need for a donor DNA template. And it works perfectly on cells that aren't dividing, like neurons or liver cells, which was a huge limitation of the old CRISPR systems.
Is it perfect, though? Because usually when you start messing with this level of chemistry, there are some kind of side effects. I saw a note in the research about something called bystander editing.
That has been the big technical challenge to overcome, for sure.
Yeah.
Think of the deaminase enzyme as a pencil eraser. But the eraser is kind of fat.
Okay.
It doesn't just touch one letter. It covers a window of about, say, 10 letters or 10 base pairs.
So if I want to change one specific C to a T, but there's another C right next to it within that window.
The eraser might smudge both of them. You might change both. You might turn the word success into sudus when you only wanted to fix one letter. That's the bystander effect.
Yeah.
You edit an innocent bystander base.
So how do they handle that? Do they just cross their fingers and hope for the best?
No, they engineer their way out of the problem. That's what's been happening over the last few years. They've developed all these new variants. One that's mentioned in your notes is TUDA-NW1, where they've physically truncated the enzyme. They literally cut off the parts of the protein that weren't necessary for the core function, which narrowed that editing window down from 10 nucleotides to just 4. They effectively sharpened the pencil.
Okay, so that makes a lot of sense. Let's look at where this is actually being used, because the theory is fantastic, but the Verve Therapeutics data is what really, really caught my eye. This is the cholesterol shot.
Yes.
And this
Yeah.
feels different because, unlike a lot of the early gene therapies, this is not for an ultra-rare disease.
This is the flagship for base editing. No
Yes.
question.
Yeah.
I mean, cardiovascular disease is the #1 killer on planet Earth. A huge driver of that is LDL cholesterol, the bad cholesterol. Verve is targeting a gene called PCSK9.
And PCSK9 is basically a keep cholesterol high gene. Is that a fair way to put it?
That's a very fair way to put it. The PCSK9 protein's job is to degrade the receptors on your liver cells that clear cholesterol from your blood.
So more PCSK9 means fewer receptors.
And your cholesterol stays high. So the logic is, if we can just break the PCSK9 gene, your liver will sprout more receptors and get much better at clearing cholesterol from your blood.
We already have drugs for this, right?
We do. We have antibody drugs, but you have to inject them every 2 weeks or every month for the rest of your life. Verve's idea was, let's just change one single letter in the DNA of the liver cells and turn off that gene forever.
One shot, a permanent fix. That's the one and done paradigm that everyone's chasing.
Exactly. So look at the timeline. Back in 2023, they ran the Heart-1 trial. They used their first-generation drug, which was called VRV-101.
And it worked.
It worked spectacularly well.
Right.
They saw a 55% reduction in LDL cholesterol. That is a massive effect. That beats most daily statin regimens by a long shot.
But the stock tanked after that readout. I remember that.
It did. It really did. Because they saw some safety signals. Specifically, they saw spikes in liver enzymes, which can be a sign of liver stress.
So the edit wasn't the problem.
No, the editor was fine. The issue was the delivery vehicle. They were using a type of lipid nanoparticle, an LNP, that caused some temporary transient stress to the liver.
Which is a no-go.
It's a no-go for a disease that isn't immediately fatal. You know, people live with high cholesterol for decades. The safety bar has to be pristine. You can't risk putting someone in the hospital to prevent a potential heart attack 20 years down the line.
Right. So fast forward to April 2025. The Heart-2 data, this was the big redemption arc for them.
Total redemption. They switched to their new drug, VERVE-102. It's the same editing mechanism, still an adenine base editor, but they used a much more advanced GalNAc LNP delivery system.
Okay, GalNAc LNP. Lots of acronyms.
We can get into the nitty-gritty later, but basically, GalNAc is a sugar molecule that acts like a VIP pass for the liver. It makes the delivery vehicle incredibly efficient at targeting only liver cells, which allows for a much, much lower dose.
And the numbers, how did they look?
Fantastic. A 53% mean reduction in LDL, with some patients in the trial hitting nearly a 70% reduction. And crucially, the safety data was clean. No scary liver enzyme spikes. It looked great.
I want to just pause on the implication of that for a second. We are talking about a preventative genetic edit for a common condition. This isn't for the boy in the bubble. This is for, well, this is for my dad. This is for millions and millions of people.
And that's why you're seeing those market projections in the report that are just astronomical. They're talking about $28 billion by 2035. We are witnessing the shift from fixing rare genetic flaws to optimizing human health on a massive scale to prevent disease before it even starts.
But the pencil base editing, it has a limit, right? You said it can do C to T and A to G. That's only 4 of the possible letter swaps.
Right, those are called transitions.
But there are 12 possible swaps you might need to make in the genetic alphabet. What if I need to turn an A into a T or a C into a G?
The pencil can't do that. It's just chemically impossible for those deaminase enzymes. For that kind of edit, you need the word processor.
Prime editing.
Prime editing.
Also from David Liu's lab. They are busy over there.
Unbelievably prolific. Yes, this was first published in 2019, but it's really hitting its stride clinically in just the last 18 months or so. Prime editing is a true search and replace function for the genome.
Search and replace.
It can perform all 12 of those base-to-base conversions. It can precisely insert a new sequence of letters. It can precisely delete a specific sequence. It is the Swiss Army knife of gene editing.
How does it work? If base editing uses chemistry, what does prime use?
It uses a fascinating fusion. It's the Cas9 nickase, so again, no double-strand break, fused to an enzyme called reverse transcriptase.
Reverse transcriptase. Now wait, that's what retroviruses like HIV use, right? To turn their RNA genome into DNA so they can hide out on our cells?
Exactly the same enzyme. We've co-opted a viral tool for our own purposes. And the real genius of the system is that they use a special engineered guide RNA. It's called a PEG RNA, a prime editing guide RNA.
And what's so special about it?
This RNA has 2 jobs. First, it has the sequence to find the right address in the genome, just like normal CRISPR. But second, it has this little tail, an extension, that actually holds the template for the new edit you want to make.
So the instructions for the new text are physically attached to the search tool.
Precisely. The Cas9 nicks one strand of the DNA, creating a little flap of loose DNA. The reverse transcriptase enzyme then grabs that flap and uses the pegRNA's tail as a blueprint to literally write the new sequence directly onto that DNA flap.
It's retyping it.
It is retyping it letter by letter right there at the site. Then the cell's own machinery sees this newly written flap and incorporates it permanently into the strand.
And because it's writing it fresh, you can write whatever you want.
Anything. You aren't limited by the narrow chemistry of a deaminase enzyme, any letter to any other letter.
And this is not theoretical anymore either. Late 2025, Prime Medicine, the company commercializing this, released the first human data on a disease called chronic granulomatous disease, CGD.
And this is a nasty, nasty disease. It's a deficiency in a protein called P47-FOX. And what that means is your white blood cells, your neutrophils, they essentially lose their ammunition.
They can't fight infection.
They can't produce the respiratory burst, this cloud of superoxide that's needed to kill bacteria and fungi. So these patients suffer from chronic, severe, often life-threatening infections. They can get fungal pneumonia just from breathing air that you or I would be completely fine in.
A terrible condition. And the trial results.
They treated patients with their therapy, called PM359. It's an ex vivo therapy, so they take the patient's own hematopoietic stem cells out of their body, edit them in the lab with prime editing, and then infuse them back in. And within 1 month, they saw restored NADPH oxidase activity. The enzyme was working. The white blood cells were armed again.
What about clinically? Did the patients actually get better?
They did. The report said the patients cleared their chronic infections. The debilitating colitis, the inflammation in their gut that usually plagues them, improved dramatically.
So search and replace works in humans.
It works. And just like base editing, so far it appears to be remarkably safe. No off-target double-strand breaks have been reported.
I really want to look at sickle cell disease here because this seems to be the battleground where all these different technologies are fighting it out. We had Casgevy approved a couple of years ago. That was classic CRISPR 1.0.
Right. And Casgevy is an absolute triumph of medicine, make no mistake. But if we look at it like engineers, it's a bit of a hack.
A hack? How so?
Well, it doesn't actually fix the sickle cell mutation. Not at all.
Wow.
What it does is it breaks a different gene called BCL11A.
What?
BCL11A's job is to act as a brake. It stops your body from making fetal hemoglobin. That's the type of hemoglobin you had in the womb, which doesn't sickle. So Casgevy uses the CRISPR 1.0 scissors to just smash the brake.
So your body says, okay, brake's broken, and turns on the backup system, the fetal hemoglobin.
Correct. It doesn't fix the underlying problem. It just enables a workaround. Now, compare that to what a company like Beam Therapeutics is doing with base editing in their Ristocil trial. The update just came out in early 2026.
And they aren't just smashing the brake.
No, they're doing something much more elegant. They are using base editing to make very precise single-letter changes in the promoter regions of the hemoglobin genes. They're basically mimicking a naturally occurring benign condition that leads to high fetal hemoglobin. They are fine-tuning the system, not smashing it.
And the results of that.
An editing efficiency of 72.8%. That is just astronomically high for a gene therapy. Usually you're thrilled if you get 30 or 40%.
And the clinical outcome for the patients?
0 vaso-occlusive crises. Those are the pain crises that define the disease. And they had sustained fetal hemoglobin levels over 60%. It's a cleaner, more efficient, and potentially safer way to get to the same result as Casgevy.
And so what's the promise for prime editing in sickle cell?
Prime is the end game. It's the holy grail for sickle cell. The mutation that causes the disease is a single-letter swap. An A gets turned into a T in the beta-globin gene. 1 wrong letter out of 3 billion.
So prime can just flip it back.
Theoretically, yes. Prime editing can just go in, search for that 1 wrong T, and replace it with the correct A. No backup systems, no workarounds, just restore the wild type, the perfectly normal, healthy gene. That's what's in the early pipeline now, and that's the ultimate cure.
It's just incredible to watch the iteration speed here. We went from the Model T, which was Casgevy, to the Ferrari, which is prime in, about 6 years?
The pace is breathtaking.
But there is this huge bottleneck that keeps coming up again and again in the notes. You can have the best word processor in the world, but if you can't get into the nucleus of the right cell...
It's useless. The delivery problem. The bio barrier. This is the biggest challenge in the entire field.
Let's talk about the two main contenders, LNPs versus AAVs, because this seems to be the dividing line between what we consider easy diseases to treat and the really hard ones.
Right. The liver is the low-hanging fruit. It's the easy target, and that is entirely because of lipid nanoparticles, or LNPs.
These are basically just tiny balls of fat, right?
That's a great way to think of it. They are tiny, engineered spheres of lipids that can encapsulate the CRISPR machinery, usually as messenger RNA, inside a protective shell.
So why does the liver love them so much? Why is it the easy target?
It's a happy accident of biology. When you inject LNPs into the bloodstream, they immediately get coated in a protein called apolipoprotein E, or ApoE, which is just floating around in your plasma.
Okay.
And your liver cells, the hepatocytes, have receptors on their surface that are specifically designed to suck up ApoE because the liver uses it to manage cholesterol metabolism. So the liver cell sees the LNP coated in ApoE and thinks, yum, cholesterol, and just gobbles it up.
So the liver actively invites the drug in. It's a Trojan horse, but the city opens the gates willingly and throws a parade for it.
That is the perfect analogy, and that's why Verve's cholesterol drug works so well. That's why we can treat other liver diseases like amyloidosis. But
Yeah.
what if you want to edit the brain or the heart or your muscles?
The liver eats it all before it can get there.
Pretty much. LNPs are notoriously bad at targeting any tissue except the liver, although many, many smart people are working on changing that. So for those other tissues, we still have to rely on AAVs.
Adeno-associated viruses.
Right. These are harmless viruses that have been stripped down to act as delivery trucks. They are very good at infecting cells like neurons or muscle cells without actually causing disease.
But the note here says AAVs have a serious cargo limit.
They have a tiny trunk. The viral shell, the capsid, can only hold about 4.7 kilobases of DNA. And here's the problem. CRISPR machinery is fat.
Huh?
A standard SpCas9 protein is big. Now add a deaminase to it for base editing. It gets bigger. Add a reverse transcriptase for prime editing. It's huge. It literally does not fit in the truck.
So how do you get a sofa into a Mini Cooper?
You use what they call the IKEA method, split Cas9.
You're kidding? You saw the furniture in half.
You literally split the genetic instructions for the protein in half. You package the front half of the editor's DNA into one virus and the back half into another virus. Then you inject both viruses into the patient.
That sounds incredibly unlikely to work. They just have to randomly find each other inside the correct cell.
They do. And they use these amazing molecular Velcro pieces called inteins. The cell reads the DNA from both viruses and builds the two protein halves separately. But as soon as those two halves bump into each other in the soup of the cell, the inteins recognize each other and snap them together to form the complete working machine.
That is wild, wild engineering.
It's incredibly clever, but it's also very inefficient. You need two different viruses to successfully infect the same cell at roughly the same time. That's why there's this massive push in the field to find smaller orthologs. Just versions of Cas9 that have been discovered in other species of bacteria. We found CRISPR systems in thousands of different microbes. Some of them, like systems called Cas2a or Cas-X, are naturally much smaller than the standard Cas9. They can fit in one truck.
Yeah.
That's where the field is heading. Finding smaller, tighter editors that don't need the IKEA treatment.
I do want to highlight one delivery story that was in the notes that felt like it was straight out of science fiction. Cancer Pam. This isn't about size, it's about logic.
Oh, this is fascinating stuff. This is a multi-omics approach.
Okay.
It addresses the big problem of off-target effects in a totally new and brilliant way.
Okay, explain the concept to me.
So, tumors have mutations. Their DNA has typos that your healthy cells don't have. They're unique identifiers. Cancer Pam designs a CRISPR system where the guide RNA specifically targets that tumor-specific mutation. It uses the typo as the trigger.
So the drug is floating around the whole body, but it only activates if it sees the tumor-specific typo.
Exactly. It scans the genome of every cell it enters. If it finds the healthy, normal sequence, it stays dormant. It does nothing. If it finds the mutated sequence in the tumor cell, it activates. And here's the clever part. Instead of cutting the DNA, it's engineered to deliver pro-inflammatory cytokines.
So it's not editing, it's calling in an airstrike.
That's precisely what it's doing. It's painting a giant target on the tumor, using the tumor's own mutation against it, and screaming to the immune system, "Hey, the bad guys are over here."
It remodels that immunosuppressive microenvironment I've read about.
It completely flips it. Tumors are famously good at telling the immune system to go away and leave them alone. This system screams, "Come get me." It's a brilliant convergence of gene editing and immunotherapy.
That is brilliant.
Yeah.
But let's zoom out again. Everything we've discussed so far, base editing, prime editing, it's all about changing a few letters, a typo here, a sentence there. But what if the whole gene is just completely trashed? What if you have a disease like cystic fibrosis?
That is the heterogeneity problem, and it's a huge one. In cystic fibrosis, there are over 2,000 known mutations in the CFTR gene that can cause the disease. You can't design 2,000 different prime editors. It's medically and economically impossible.
So you don't want to just fix the typo, you want to replace the entire corrupted chapter of the book.
Right. You need a select all delete paste function. Prime editing is too small, too delicate for that. And so now we're entering the era of what people are calling CRISPR 3.0 large-scale rewriting. The systems leading the way are called passage and paste.
Passage. Okay, let's break that down. It's an acronym.
It is. It stands for Prime-Assisted Site-Specific Integrase Gene Editing.
That's a mouthful.
It is, but the concept is simpler. Think of it as drag and drop for genes. They use prime editing not to fix the gene, but to install a tiny landing pad in the genome. It writes a specific sequence called an @TB site. It's like installing a universal USB port into a specific safe spot on the chromosome.
A universal port. I like that.
Exactly. Then, in the second step, they introduce a different enzyme, an integrase, which is derived from phages. Those are viruses that eat bacteria. And this integrase enzyme is programmed to recognize that USB port and drop a massive payload right into it.
And by massive payload, you mean entire genes.
Entire genes. We're talking kilobases of DNA, a chunk of code big enough to contain the entire, healthy, full-length version of the CFTR gene, for example.
So step 1, prime edit the landing pad in. Step 2, the integrase drops the healthy cystic fibrosis gene right onto that pad.
Precisely. And the most recent data on this, specifically a system they call ePassage, published in 2025, showed a 30% integration efficiency in human cells.
And is 30% good?
It's incredible.
Yeah.
For a disease like cystic fibrosis, correcting just 10% or 15% of the relevant lung cells is thought to be enough for a functional cure. 30% is a home run.
So we have pencils for small changes, that's base editing. We have word processors for search and replace, that's prime. And now we have cargo ships for whole gene replacement with passage.
And there's one more tool in the kit that's important, the volume knob.
The volume knob.
Epigenome editing. This is important because, as you know, sometimes you don't want to change the hard drive, you just want to change the software.
Right. The DNA is the hardware. The epigenome is the layer of software on top that tells the cell which genes to run and how loudly.
Exactly. Sometimes you have a perfectly good gene that is just turned off when it should be on, or vice versa. Or think about treating something like chronic pain.
Yeah.
You don't want to permanently break your pain receptor genes. You might need them later if you touch a hot stove.
You just want to mute them for a while.
You want to silence them temporarily, like taking an aspirin that lasts for months. So for this, they use what are called dead Cas9, or dCas9.
Yeah, Cas9.
It's a zombie protein. It still uses the guide RNA to grab onto the DNA at a specific address, but it doesn't cut at all. It can't. It just sits there. But they attach it to another enzyme, like a methyltransferase.
And methylation is the chemical tag that usually means off, right?
Yes. It's like putting a do not disturb sign on that gene. The cell's machinery sees the methylation and just stops reading that gene. They're looking at this for Alzheimer's, to silence the tau gene, which forms the tangles in the brain.
Or you can do the opposite, I assume. Turn things back on.
Right, which is the approach for Fragile X syndrome. In Fragile X, a critical gene called FMR1 is incorrectly silenced, turned off by mistake. So they can use dCas9 fused to a demethylase to go in and strip those silencing marks off and turn the gene back on.
And since it's not cutting the DNA at all, I have to assume the safety profile is even better.
Much better. No risk of breaks, no risk of translocations. And if you take it one step further and do RNA editing using a different enzyme, Cas13, to target the temporary message instead of the permanent DNA, it's fully reversible.
Yeah.
If you stop taking the drug, the effect vanishes. It's perfect for acute conditions or for testing if a therapy will work before you commit to a permanent, irreversible DNA edit.
It really feels like the toolbox is full now. We can cut, we can write, we can replace, we can silence, and we can boost. But none of this matters if the regulatory bodies, if the FDA says no. And that seems to be the other massive shift that happened in late 2025.
The plausible mechanism pathway. This is a case of regulatory innovation finally catching up to the pace of scientific innovation.
And this was triggered by these N of 1 cases, right? These one-person clinical trials.
Yes. The most famous one, the one that really forced the issue, was the infant with CPS1 deficiency. This is a super, super rare metabolic disorder. Ammonia builds up in the blood. It's usually fatal within months of birth. There are maybe a handful of cases a year worldwide.
So you can't run a phase 3 clinical trial with 500 people if
Yeah.
there aren't 500 people with the disease on the entire planet.
Exactly. So historically, these kids they died. There was no path forward. But in this one case, a team of researchers went from diagnosis to designing a custom CRISPR treatment in 6 months. They designed a guide RNA that was specific to that one baby's unique mutation.
And it worked.
It worked. The baby survived. And the FDA looked at that, and they had a moment of realization their old rules, which required these massive randomized trials, were effectively a death sentence for patients with ultra-rare diseases.
So they changed the rule.
They did. They are now allowing, for these ultra-rare cases, an approval based on biomarker endpoints, which means if you can prove the edit happened, look, the DNA is changed, and you can prove the intended effect is happening. Look, the toxic ammonia is gone from the blood, and the mechanism of action is well understood and plausible. They will consider approval without years and years of efficacy data.
That is a complete game changer for the rare disease community. It just opens the floodgates.
It does. But it also brings us to the elephant in the room.
The costs.
The cost. The report says the gene editing market is projected to hit $28.77 billion by 2035. But if these are one-and-done cures, how on earth do you price them? We saw this a few years ago with Hemgenix for hemophilia. It was priced at $3.5 million for one dose.
The argument being it's still cheaper than a lifetime of care.
That's the tension. If I cure your hemophilia with one shot, I save the insurance company, say $20 million over your lifetime in factor replacement therapy. So from their perspective, $3.5 million is a bargain. But do they want to write me a check for $3.5 million up front today?
It's the mortgage model for healthcare.
That's exactly where we're headed. We're going to see a shift to annuity payments. The insurer pays the drug company $200,000 a year for 15 years, but only as long as the cure continues to work. It aligns the incentives.
But as we move from rare diseases to common ones, like cholesterol...
The math gets much harder.
Right, because the alternative is a generic statin that costs $5 a month. Who gets the super pencil then?
Who gets the super pencil and who pays for it?
We've covered the tech, the delivery, the regulations, the money. I want to end on the thought experiment that was mentioned in the conclusion of the source material. It's about the germline.
The third rail of genetics. The forbidden territory.
We've been talking exclusively about somatic editing, fixing my liver, my blood. That edit dies with me. My kids don't inherit that fixed PCSK9 gene.
Correct.
But if prime editing is as safe and as precise as the 2026 data is starting to suggest,
then the arguments against editing embryos start to crumble. In 2018, when the He Jiankui scandal broke, the scientific community agreed on a moratorium because the tools were too blunt. The argument was, don't edit embryos because you might make a mistake and break the baby. It was fundamentally a safety argument.
But what if the risk of that mistake, of an off-target edit, drops to near zero? If prime editing allows you to cleanly change a Tay-Sachs gene back to a healthy gene without touching anything else in the genome,
then the moral calculus shifts entirely. The question becomes, if you can safely prevent a child from being born with a horrific, painful, and fatal disease, is it unethical not to do it? The pressure from parents to fix the embryo before it's even implanted becomes overwhelming.
and then inevitably you slip from fixing a fatal defect to just reducing risk.
Right. We know of a rare, naturally occurring mutation on the APP gene that confers powerful protection against Alzheimer's disease. It's a single-letter change.
So if you're a couple going through IVF and the clinician says, for an extra fee, we can safely use prime editing to give your future child this protective gene,
Do you do it?
most parents would say yes in a heartbeat.
Of course they would. And what about the gene variant associated with needing less sleep or the one for higher muscle density? The line between therapy and enhancement is going to blur so fast because the tool, the prime editor, it doesn't know the difference. The tool just writes whatever code you tell it to write.
We saw in the notes that they are already using these tools for genetic sexing in mosquitoes to control malaria-carrying populations. We are literally rewriting the code of entire species in the wild to suit our needs.
We are taking the controls away from evolution. For 4 billion years, life on this planet was driven by two forces, random mutation and natural selection. It was a slow, blind, and often very cruel process.
And now,
For the first time, it is intentional design. We are seizing the pen from nature. And that is the most profound shift in the history of biology. And we're living through the first paragraph of that new chapter right now.
it's a lot to process. If you want to follow the money and the science on this. The advice is to keep an eye on Beam Therapeutics and Prime Medicine later this year. The BLA filings are coming, and those regulatory decisions are going to set the precedent for everything we just talked about.
The next 12 months will dictate the next 50 years of medicine. No exaggeration.
Massive thank you for walking us through this whole stack today. This was dense, but I feel like I finally truly understand what CRISPR 2.0 actually means.
It was my pleasure. Always is.
And to you listening, stay curious. We'll see you in the next deep dive.